Hereditary information is encoded in the chemical language of DNA. In this lecture, we will review the structure of DNA and find out how it is replicated. Do you remember which monomers compose nucleic acids such as DNA? Yes, nucleotides, which are composed of a phosphate group, a pentose sugar, and a nitrogenous base. And if you will recall, the pentose sugar has an orientation based on its carbons. The 3' carbon and the 5' carbon are on opposite sides of the nucleotide molecule. The 5' carbon is closest to the nucleotide's phosphate group. And to form nucleic acids, we have to connect these monomers together. The bond that forms between the nucleotides is a covalent bond specifically called a phosphodiester bond. It occurs between the pentose sugar of one nucleotide and the phosphate of another nucleotide. The sugar and phosphate form the sugar-phosphate backbone of the nucleic acid. The bases are not part of the backbone and can form bonds with other nucleic acid strands. Nucleic acid strands such as DNA run in a 5' to 3' direction based on the sugar orientation. DNA is double-stranded, so the other strand must run in the opposite direction for the bases to properly interact. We say the strands run anti-parallel to one another. There are 4 different types of nucleotides based on what kind of nitrogenous base they have. We commonly call different nucleotides bases. Adenine and guanine have nitrogenous bases that are purines. Purines have 2 rings. Adenine has no oxygen attached to its rings, while guanine does. Thymine and cytosine have nitrogenous bases that are pyrimidines. Pyrimidines have only 1 ring. Thymine has 2 oxygens attached to its ring, while cytosine has only 1. These bases can pair with each other, but only in very specific combinations according to Chargaff's rule. Chargaff discovered that DNA contained the same amount of adenine as thymine and the same amount of guanine as cytosine. He concluded that adenine paired with thymine and guanine paired with cytosine. This is due to a few reasons. The first is spacing. A purine-purine pair would be too wide and couldn't bind due to the overlap. 2 pyrimidines would not come close enough to each other to form bonds. But a purine-pyrimidine pair is just right to form bonds. And what are those bonds? Hydrogen bonds. But these are also specific. Adenine and thymine form 2 hydrogen bonds, while guanine and cytosine form 3. So an incorrect pairing would not form the correct number of hydrogen bonds, and it would have the wrong spacing. Exactly how does the phosphodiester bond between nucleotides form? In reality, the nucleotides used to build DNA have 3 phosphate groups. These molecules are called deoxyribonucleoside triphosphates, or DNTPSs. The N stands for the base and can be adenine, thymine, cytosine, or guanine. Notice how this molecule looks similar to ATP due to the 3 phosphates. That is because it is similar. The multiple phosphates on the molecule store energy in their bonds, just like ATP. Forming a phosphodiester bond is an endergonic reaction. When a DNPT is added to DNA, the 2nd and 3rd phosphates are cleaved from the molecule. There is a release of energy, which is used to form that phosphodiester bond. Please be aware that while DNA is frequently presented in this ladder diagram, with the 2 antiparallel sugar-phosphate backbones as the sides and the nitrogenous bases as the rungs, the molecule is really twisted into a double helix, and the phosphates, sugars, and bases are really composed of atoms. So this space-filling model shows a more realistic relationship of the positions of the atoms in the DNA molecule. However, we will be using the ladder diagram in most representations. You learned in the very first unit that DNA carried information in the cell. So how does it do that? Well, information is encoded in the nucleotide sequence. A wide variety of information can be encoded because the 4 different nucleotides can be used for each space in the DNA instructions. For example, if we just looked at a 10 nucleotide sequence, we could place one of 4 nucleotides in each of those 10 spots, resulting in over 1 million different possible combinations. But organisms contain much more than 10 nucleotides in their genomes. If we look at humans, we find over 3 billion base pairs. The possible combinations would be 4 with an exponent to 3 billion. Now, in reality, Not all those nucleotides code for cellular products, but it is still a jaw-dropping number of combinations.